Thank you for tuning into my talk. My name is Brittany Hoelscher, and today I will be discussing my work on the functional roles of the gap junction gene innexin in the freshwater snail Lymnaea stagnalis. In an electrical synapse shown here, one neuron is bound to another via protein called gap junctions that form channels to allow the direct flow of electrical signals, ions, and small molecules from cell to cell. In invertebrates, the gene that codes for the gap junction protein is named innexin. Because there is limited information on the innexin gene, our lab asked the question, what are the functional roles of innexin? Based on previous research in other invertebrates, we hypothesized that innexin would play a role in many behaviors, but we wanted to specifically focus on development. To do this, we utilized the central nervous system of the invertebrate snail Lymnaea stagnalis shown here. With our snails, we can dissect out the CNS and even isolate single neurons to study at a single cell level, a feat that is extremely difficult to achieve in mammals. Our lab was the first to identify the sequences of the 8 innexin paralogs present in our snail's genome. Here, we have a multiple sequence alignment of the 8 innexins we identified. Once we had the sequences of our snail innexins, we dove into our hypothesis that innexins are important genes regulating development. To test our hypothesis, we performed RNA sequencing on 3 age groups of snails: 3-month-old young snails, 6-month-old adult snails, and 18-month-old aging snails. You can see from our RNA-seq experiments that 3 innexin paralogs were identified as having differential expression between all ages. Innexin 1, innexin 4, and innexin 5 all had significant increases in their expression between young and adult snails, which slightly dropped from adulthood to aging snails. These findings were confirmed via qPCR. Our RNA-seq results indicate that while innexin genes are involved in changing in response to normal development of Lymnaea, their role may be more critical during adulthood and into the aging process. Other genes then, perhaps those related to chemical synapses, might be more important in the earliest stages of development. The question we next want to ask is, what happens when innexin genes are no longer functioning? Does development become abnormal? To answer this question, we are designing CRISPR-mediated knockouts to mutate innexin genes in Lymnaea, in which mutational RNA is injected directly into a snail at the 1-stage level, then the snail is grown to adulthood. The innexin knockout snails are then monitored to check for abnormal changes in developmental characteristics. With that, I would like to acknowledge our funding sources and thank you for listening.